Okay.
Cool. So maybe I'll start so that more people are joining. Hi, good morning, everyone. So before we start, I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining me from today. I pay my respect to their elders, past, present, and emerging, and to the Aboriginal elders of other communities. So welcome everyone. My name is Anya and I will be chairing the seminar today. I am a PhD student at the CCMP Center at the Monash Node. And before we start and introduce our speaker, I would like to quickly go over some housekeeping rules. So first and foremost, I'd like to let you know that today's seminar will be recorded and you will be able to find this as well as previous recordings on the CCMP website. And the presentation today by our speaker will be 45 minutes, followed by 10 to 15 minutes Q&A session. So if you have any questions, you can leave them in the chat box till the end of the seminar or by raising your hand so we can all address the questions that are coming. Please also make sure that your microphone is muted throughout the presentation. So our speaker today is Assistant Professor Rie Nygaard. And Rie is an assistant professor at Weill Cornell Medicine in New York City, USA. She earned her PhD from the University of Copenhagen in Denmark, focusing on structure and dynamics of G protein-coupled receptors. She then completed a postdoctoral fellowship at the Stanford University, followed by another postdoc and a research associate position at Columbia University in Filippo Mancia's lab. In the Mancia lab, she worked on determining the structure of several small membrane proteins using single particle cryo-EM, and she will present some of that work here. In her lab, she is developing a research program focused on understanding the molecular effects of radiation therapy, focusing on targets that are regulated or modified as a response to radiation therapy. So her talk today would be about structural insights into glycosyltransferase function. And this is basically something that catalyzes the addition of sugars to diverse substrates. And when it's embedded in or associated with the membrane, these enzymes often rely on polyisoprenyl-phosphate or pyrophosphate lipid carriers to transfer glycan moieties. So here Rie will present different bacterial GT structures and compare the substrate binding mechanisms. Additionally, she will present how they successfully visualized how the GtrB enzyme transitions during the catalytic cycle. So using time-resolved cryo-EM, multiple distinct conformational states and the initial substrate-bound state were identified. So thank you so much for joining us here, Rie. And over to you. We're lucky to have you here. So the screen is all yours.
Thank you. Can you see my screen?
Yeah. Yeah.
Yeah. Okay, great. My name is Rie, and I am, as you said, an assistant professor, newly hired here at Weill Cornell Medicine. I was previously in Filippo Mancia's lab at Columbia University, and the work I'm going to be talking about here today is from my time in Filippo's lab. I was hoping when I got the invitation back in December last year, I think that was it, that I would be able to present some of the work we're doing in my lab here. But unfortunately, I just don't feel like it's mature enough to present on for 45 minutes. So what is going on here? Oh, here we go. All right. So just wanted to, even though I'm not going to present the work, wanted to give a short introduction about who we are and what my plan is for the lab. So I'm building a structural biology lab at Weill Cornell Medicine, where we're going to look at different targets that are affected by radiation therapy. So we have some targets that change conformation based on a reactive oxygen level in the cell, and also some targets that are upregulated in response to radiation in the immune cells. And currently, I have two people in the lab. So one is Yachi Liu, who was also in the Mancia lab but followed me here. So I'm very happy to have her here. She's a research associate, and then we just hired Alexander Hager, who's a research technician. But hopefully I'll have a chance to present this work to you soon. But for today, I'll focus on work I did in the Mancia lab. So, as you know, we are facing a health crisis because, or it's in the making because of antibiotic resistance. So there's a rapid increase in antibiotic resistant drugs or bugs, and at the same time, we have had very few newly developed antibiotics. Actually, there's been no new class of antibiotics over the last 30 years. So the CDC estimates that over 2 million illnesses in the US per year is due to antibiotic-resistant infections and at least 23,000 deaths. So if we look at antibiotic targets, there are several important targets, but what I'm going to be focusing on today are the targets that is found in the synthesis of the cell wall. So we already have some great antibiotics that are targeting the cell walls, such as the beta-lactams, for example, penicillin, that binds to the penicillin binding protein that's part of cell wall synthesis. But what is important for developing a good antibiotic is that it's targeting as an enzyme that's essential for the bacterium, and also that it's unique to the bacteria. So it's important that this is an enzyme that's only present in bacteria or is significantly different in eukaryotic cells, which is why the bacterial cell wall is such a good target, because it really does look a lot different from the eukaryotic cell wall. So if we take a look at what the bacterial cell wall or envelopes look like, we can divide them into three groups. There's the mycobacterium cell wall, which is has a very thick cell wall with multiple layers, peptidoglycan, arabinogalactan, and mycolic acid layers. This gives the mycobacterium a great protection, and also it makes them resistant to a lot of antibiotics, which is a big problem. Then we have the gram-negative or gram-positive bacteria that also have a very thick cell wall with a thick peptidoglycan layer and a layer of teichoic acid as well. And then the gram-negative bacteria that have two plasma membranes, or the membranes, so the inner and the outer membrane, and in between the two, we have this peptidoglycan layer that gives the cells rigidity. And I also want to mention here, because I'll come back to this, that gram-negative bacteria have these lipopolysaccharides on the outer leaflet of the outer membrane. So what's common for all of these cell walls is that they are mostly composed of sugars and small peptides that are linked together. And for this, for the cell wall to be synthesized, it's important. One group of enzymes important, and that's the glycosyltransferases. So we can divide glycosyltransferases into two groups. We have the donor glycosyltransferases that transfer a sugar molecule to another molecule, such as a peptide or another sugar, or like here. And then we have the acceptor, where the sugar is transferred onto this polyisoprenyl substrate. So what I'm talking, these specific groups of, I should preface this by saying the specific group of glycosyltransferases I'm talking about today are the membrane protein glycosyltransferases. And what's common for them is that they have this isoprenyl phosphate substrate. So this is a table showing the different isoprenyl phosphate substrates from different organisms. So today I'll focus on bacteria. So for gram-positive and gram-negative bacteria, these polyisoprenyl-phosphates are generally undecaprenal pyrophosphate, so they have 11 isoprenyl groups, or pyrophosphate, where they have 2 phosphate groups here. And for Mycobacterium, it's slightly different. It's a decaprenyl phosphate, which means that it has 10 isoprenyl groups. But other than that, they are very similar. So if we're now going to focus a bit on Mycobacterium. So I'm sure you know that tuberculosis is a very deadly disease. It's actually the deadliest infectious disease in the world at the moment. Um, it's very contagious, and transmit through the air, and it's caused by the Mycobacterium tuberculosis bacteria. So there are over 10 million new cases per year and over 1.2 million deaths. And this first couple of stories was spearheaded by Yachi when she was a postdoc in Filippo's lab, and is where she solved the structure of 2 of these glycosyltransferases from Mycobacterium. The first is PIM-E, which catalyzes the transfer of the 5th PIM to this PIM layer between the plasma membrane and the peptidoglycan layer, which is an essential layer for the survival of the Mycobacterium. And then also the structure of the of the AFTB, which is an arabinogalactam enzyme. It helps the catalysis of a reaction to form the arabinogalactan layer. And if we take a closer look, so you can see both. Yeah, I wanted to point out that both of these enzymes were solved with a Fab bound to increase the size of the particles, but also to give it some shape. So these are the 2 structures. They both fall in the GTCA glycosyltransferase family. And you can see for the AFTB, it has 11 transmembrane helices, and for PIM-E, it has 12 transmembrane helices. And characteristic for this class of enzymes is that between TM1 and 2, they have this long loop with 2 juxtamembrane helices and the active aspartate at the tip of JM1. What's also interesting is that for these 2 structures, we see the substrate bound, and you can see that it snakes its way along the transmembrane helices. You can't see the full substrate, but it snakes its way here, so the substrate can come in from the membrane to reach the active site of the enzyme. But yeah. So moving on to gram-negative bacteria. So just reminding you that gram-negative bacteria like E. coli are rod-shaped, have these rod elongated shape. And the cell wall of gram-negative bacteria consists of an inner and outer membrane. And in between these 2 is the peptidoglycan layer. So the peptidoglycan layer consists of polyglycans, and they are modified with a pentapeptide, and these pentapeptides are then linked together to form this highly rigid polymer that can preserve cell morphology and protects the cells from environmental stress. And it is an ideal target for antibiotics because this peptidoglycan layer is unique for bacteria, and it's not found in eukaryotic cells. So there are 2 complexes that are important for the formation of the peptidoglycan layer in gram-negative bacteria. One is important for division, and the other is important for elongation. So the division is called the divisome, and the elongation is called elongosome. And for this story, I'll focus on the two central players of the elongosome, and that is Rod A and PBP2. These are the enzymes important, critical for the synthesis of peptidoglycan. So these two enzymes, they form a complex. So Rod A is here in green, and PBP2 is penicillin binding proteins, also what um, and um, Rod A is glycosyltransferases. It catalyzes reaction of lipid 2. I'm showing it here. The lipid 2 consists of an endocaprenyl pyrophosphate with two sugars attached. Let me see if I can point. Two sugars attached, and then this pentapeptides modification. And what this enzyme does, it links the already existing peptidoglycan layer with this incoming lipid 2. And as a byproduct, you have this endocaprenyl pyrophosphate. This glycan chain then moves up the enzyme like this until it reaches the transpeptidation site. And where PBP2 then catalyzes reaction of transpeptidation, basically linking the two pentapeptides together. So after we started this project, another structure was published of the T. thermophilus E. coli, not E. coli, the T. thermophilus Rod A PBP2. But we still had a number of questions. For example, where does the substrates bind? And how does this enzyme work? How is it moved from the transglycosylation site up to the transpeptidation sites? And what residues are involved, and what movements of the proteins are involved? So to answer some of these questions, we set out to determine the structure of the E. coli Rod A PBP. And to do this, we created a fusion between the Rod A protein and PBP2, so linking the C terminal of Rod A to the N terminal of PBP2. And we purified it using nickel purification and reconstituted it into nanodisk. And here are some 2D class averages of this. As you can see here in green, no, whoops. Okay. In green, we have Rod A, and in blue, we have PBP2. Okay, here we go. Spins. So here you can see Rod A is in rainbow, and the gray helix is PBP2. So PBP2 only has one transmembrane helix. And this is just a map where we did a local refinement of the transmembrane region of the protein. This also showed that Rod A had GTCB fold. And what's characteristic for this is that there's this large loop between TM7 and 8 with these juxtamembrane helices. So we basically have a tight helical bundle with all of these transmembrane helices, and then one TM7 extending away from this bundle. So if we take a closer look, we're now looking for where the ligand would bind. We can see that there are two main cavities in the proteins, and these are all both aligned with highly conserved residues. So this is colored in conservation. And just to remind you, the reaction is that lipid 2 is linked to, these two sugars are linked together, forming like longer and longer glycan chains. But the question is then, which is the donor and which is the acceptor cavity. And based on our work, we have narrowed it down, so we think that cavity A is the donor and cavity B is the acceptor. And I will now describe why it is we think that. So in collaboration with Phil Stanfield's group at University of Warwick, we ran coarse-grained MD simulations trying to identify what sites in the protein had a high affinity for lipid 2. And what we saw is that, as we expected, cavity A and cavity B are the main sites for lipid 2, where lipid 2 likes to bind, and cavity B has much higher affinity than cavity A, based on this coarse-grained simulation, suggesting, which makes sense, that that this would be where the new lipid 2 comes in, and cavity A is basically where the undercaprenal leaves. What we saw here is that we have some very conserved arginine residues in cavity A and at the membrane interface in cavity A and cavity B, and which we propose could be important for binding to the phosphate groups of lipid 2. So if we take a closer look at these cavities and these conserved residues, we wanted to test the effect of mutating these residues to alanines. And we did this in collaboration with David Roper's group, but also at University of Warwick and his student Chris Graham. So basically here, this is lipid 2 alone without any enzyme, and this is when we add our fusion rod A PPP2. And you can see here, this is a TLC plate where we can see the polymerization of lipid 2, the products of this enzyme. But if we mutate these highly conserved arginine residues, they are pointed out their conservation. They're either arginines or positive residues. We can see we lose enzyme activity, which makes sense. So now we wanted to take a closer look and see if we could see any density for undercaprenal pyrophosphate. Oops, it's a bit slow. So basically, yes, we saw some density for undercaprenal pyrophosphate, although it's not the whole molecule and it's rather weak. We built in undercaprenal pyrophosphate just to see how it would fit. And if we compare it to the density we can get from running so we ran an MD simulation, we put in undercaprenal pyrophosphate, and this is the density over the whole simulation. We see that it matches very well with the density we see in our structure, suggesting that this density is in fact undercaprenal pyrophosphate. So to better understand what movements takes place for this reaction to proceed, we started a collaboration with Linda Columbus's group at University of Virginia and her postdoc, Megan Dufresne. So first of all, we looked at the quality of the density, and we can see that we have much higher resolution for this part of the right side of the protein, and the lower resolution, of the left part of the protein, suggesting that there might be dynamics between these two domains. And to test this, we did a DEAR experiment, so double electron-electron resonance spectroscopy. We basically mutated two cysteine residues, one in TM3 and one in TM2. And then we measured the distance between, and we labeled these cysteines with an R1 side chain, which has an unpaired electron, and we measured the distance between these two spin labels. And what we see is that we see this distribution of different distances, suggesting that there is a lot of dynamics happening between these two domains. And yes, so now we this is what we propose is the active site, and this is the active aspartate aspartate. And you can see again that if we mutate it, we completely lose activity. So what we're proposing is that this aspartate abstracts a protein from the 4' hydroxyl of lipid 2, and this abstraction of the protein then allows for the 4' oxygen to perform a nucleophilic attack. And this breaks the bond and then releases this undercoprenyl pyrophosphate and forms this new lipid 4, we call it, where there's four sugar molecules. And this is a model of how this reaction takes place. So we have two lipid 2s to begin with. They're linked together. Undercoprenyl pyrophosphate is released from the donor side, and this glycan chain starts moving up towards the transpeptidation side. And this is just a video illustrating this. And you can see this opening of the enzyme to allow for the undercoprenyl pyrophosphate to move from cavity B to cavity A to release undercoprenyl pyrophosphate. And at the end here, we have, whoops, it's a bit glitchy, but at the end, the glycan reaches all the way up to the transpeptidation sites, where it can then be linked to the existing peptidic glycan. So this was one story. At the same time in the lab, we're working on a different story with looking at LPS synthesis, the final step in LPS synthesis. So LPS is the lipopolysaccharide that is the main constituent of the outer leaflet of the outer membrane of gram-negative bacteria. And WL is an enzyme that the reaction it catalyzes is that it links the O antigen that's carried by the undercoprenyl pyrophosphate in this case to the lipid A core, which is the final step in LPS synthesis, creating a fully mature LPS that can then be transported to the outer membrane by the LPT transport system. So this is the structure of the overall structure of WL, and this, I will say, this project was spearheaded by Khurram Ashraf, also in the Mancia lab at the time. And let's see, spins. And this protein structure was also solved in complex with Fab to give it some more definition or some more features for alignment. And overall, what we see is that it also has the GTCB fold, meaning that it has this long loop between two of the transmembrane helices with lots of juxtamembrane helices. And if we take a closer look at this protein, we can see that it indeed also has two cavities. This cavity 1 is the same as cavity, similar to cavity A in rod A, and then this other cavity 2. And these cavities are also aligned with positive charged residues, arginine residues, and if you mutate these residues, you lose activity. Which was what was fun was that we were in the lab at the same time working on these project at the same time, doing these two enzymes that have very different substrates and have very different functions. But what we see is that they're actually quite similar in fold, although WL has 2 extra TM helices compared to rod A. They do have overall the same helical bundle with one TM7 or TM9 extending away from the helical bundle, and it's stabilized by this juxtamembrane helix, which has this very conserved arginine residue that's stabilizing the phosphate group of undercaprenyl. So to conclude on this, we determined the structure of two glycosyltransferases, and they both turned out to have the GTCB fold. And they both have these conserved arginine residues that coordinate the pyrophosphate groups of undercaprenyl pyrophosphate. And the dynamics of Rode PVP2 can explain how substrates move within it. Yeah, even with two very diverse substrates, we can have this very conserved fold. So the next step for the Rode project is to try to get a more complete alongosome structure, trying to see if we can pull this Rode PVP complex out of E. coli and tag the proteins endogenously, and then see if we can get a structure with more of these elongosome proteins present. And just to finish this part of the talk, I really have to acknowledge, of course, Filippo Mancia, whose lab this was done in. Kwam Ashram was the main author for the WL project. Several other people in the Mancia lab. I would like to, of course, thank Oliver Clark, which I'm sure some of you know, who's been very helpful in data processing. Then David Roper's lab at University of Warwick, Phil Stansfeld's lab at University of Warwick, Jonathan Dorkin at Columbia, Linda Columbus at University of Virginia, and Stephen Trent at University of Georgia. And I'm now going to move on to the second part of the talk, where we look at a different enzyme in GtrB. It's actually an enzyme that comes before WL in the synthesis of LPS, and that is GtrB. So the GtrB structure was solved maybe 10 years ago in the Mancia lab, and but in the apo state without a substrate. And what we set out to do in this project is to see if we can better understand where the substrates bind and the trajectory of the enzyme reaction. So we have this enzyme catalyzes the transfer of glucose from undercaprenyl or to undercaprenyl-phosphate from UDP glucose. So basically transferring these glucose to an undercaprenyl-phosphate that can then use this as a substrate for subsequent enzyme reactions. And to study this, we use time-resolved cryo-EM and flash photolysis. Yes. So the idea here was that we have a cage substrate. So we put a chemical cage on the glucose group of UDP glucose. And in this case, it's a nitrobenzyl. And when we shine UV light on this group, it detaches, and we have this functioning substrate that can then bind to the enzyme, and the reaction can take place. So in collaboration with the Stoll lab in Colorado, we designed a vitro bot where we basically plot the grids. Then they go through the vitro plot. There's a UV light that shines on it for around 17 milliseconds, and then we immediately freeze the sample. And using this method, we were hoping to capture the enzyme in a substrate-bound form and potentially different different steps along the reaction trajectory. And so this is the density of the enzyme. I don't know why it's so glitchy, but as you can see here, this is a tetramer with 2 transmembrane helices in yellow and 2 juxtamembrane helices in blue, and then this glycosyltransferase domain down here. And the structure of this tetramer was not surprising because we had a structure to begin with, but what we're trying to do now was to divide all the particles into different groups to hopefully see different steps along the trajectory. And to do this, we used 3D variability analysis in cryosparks. So let's hope this works. You can see this is divided into 10 intermediate frames. And you can see there's quite a lot of movement within the transmembrane helices. Let me see. And also some up and down movement of these juxtamembrane helices. And we're able to then, using 3D classification, to divide them into discrete classes. And what we observed was that we isolated 3 different states. One that we call the substrate-bound state, which has undercoprenyl-phosphate and the UDP glucose as 2 separate molecules with separate densities. Then we have the pre-catalysis state, that's just before the reaction takes place. And then we have also a product-bound state. And if we take a closer look at this substrate-bound state, here we can see that there's the undercoprenyl-phosphate group and the anomeric carbon of the glucose are about 10 angstrom apart, which is too far for the reaction to take place. But also interestingly, the membrane is sitting here above this juxtamembrane helices, so this undercoprenyl-phosphate has to reach quite far down into the away from the membrane to reach the active site here. For the phosphate group is stabilized by these arginine residues, and it is quite stable here. We ran MD simulation to see if it was stable in this state or if the undercoprenyl would leave the binding site or go back up into the membrane. And what we see is it's quite stable, although the glucose is quite flexible. It's not really coordinated well by any residue. We then also saw this, the pre-catalysis state, where the phosphate group of undercoprenyl-phosphate moved much further down into the active site. It's now coordinated by some arginine residues further down in the active site, and it's now in a position where it's ready for the reaction to occur. But you can see that the densities are still separated. And here's just a comparison between the substrate-bound and the pre-catalysis state. And you can see here this is the movement that takes place. If we look at the overall movement that takes place for this to happen, we notice that especially the position of the JM helices change quite a lot. So the JM helix actually moves down at the same time as undercoprenyl-phosphate, pushing the undercoprenyl-phosphate into the active site of the enzyme. And to look at this better, we ran steered MD simulations and to see what residues coordinated the phosphate group of the undercaprenal phosphate. And this is a MD simulation where we are pulling the phosphate group into the active site. And this is just this is just looking at the different arginines and lysine residues and seeing how it's first coordinated by this 125, and then it's released and then starts getting coordinated by this 197 arginine when it moves further down into the binding site. When we take a look at the product state, we see again two separate densities, but now the undercaprenal phosphate density has this large group here where we fit in the glucose, and we're left with the UDP here, suggesting that we actually are able to capture the product state. Looking back, and if we had to redo it, we would probably have waited a little bit longer before freezing, just to potentially also trap a state where undercaprenal glucose is actually leaving the active site. But what is interesting here is that this tryptophan residue swings away from this juxtamembrane helix, kind of opening a way for the product to leave the enzyme. And this is what I've tried to illustrate here. So this is the substrate-bound form. It then swings away. You can see that this tryptophan swings away, opening a route for the substrate or the product to leave the enzyme. And this is what we did here. We have this the tryptophan swinging out, and we ran another steered dynamic simulation, pulling the product out of the enzyme. And we found that we actually didn't need to use like it was negative work to for it. So the meaning that the exit of the product is spontaneous. So this is so the final model is basically we have this stabilization of the structure membrane helices when the substrate binds. They then move in concert with the substrate down, so pushing the substrate into the active site, binding different positive residues on the way. And then when the reaction happens, after the reaction happens, this tryptophan swings out, making space for this product to leave the enzyme. And for this project, I need to specifically thank Ryan and, of course, Filippo and the whole Mancia lab. And also a special thanks to Ollie again, the Stoll lab at University of Colorado Boulder, the Lowry lab in Taiwan for helping with the chemistry, and the Quick lab for some functional assays. And to conclude everything, I thought I would just give you an overview of the different donor polyisoprenyl-phosphate glycosyltransferases we've talked about. So there's the GCCA class that have these at least 7 transmembrane helices, and between TM1 and 2 have these 2 juxtamembrane helices. And there's the GTCP fold where there's another long loop between TM7 and 8. And common for these is that the substrate binds in the membrane and it never leaves the membrane. The active site is at the border of the membrane and the extracellular or the periplasmic space. Whereas for the acceptor PPGT, like GtrB, that has a GTA fold. That's a lot of acronyms. You see that the membrane, only the trans only the two transmembrane helices are in the membrane, but the substrate actually has to leave the membrane to go into the active site of this glycosyltransferase domain. So we have to create this groove that goes from the membrane all the way to the active site. And with that, I'm done.
Oh, well, thank you so much, Rie, for sharing your research. It was all very interesting and impressive. So I guess that now we can open seminar for questions. So you can either type them in the chat box or click the raise hand icon. So we're just gonna wait a bit. Maybe while we wait, I have a quick question. So you mentioned you added a fab to increase the size. So I was just wondering if this fab was designed to bind to a specific part of the protein. Is there any sort of parts of this specific protein that you find challenging that you want that fab to bind to?
Yeah, no, it wasn't as clever as that. We purified the proteins, and then we screen it against a library of fabs. Well, actually, it's the Koshakov lab in Chicago that did that part of the work. But no, we didn't.
Okay.
It wasn't. It was against the whole protein. But I mean, we would expect it to bind something that has that's outside of the membrane, of course, that has
Oh,
some features.
thank you. All right, I think Matt has a question.
Yeah, g'day. Great talk. Very, very elegant work with the GtrB project. I guess I got a few questions around that. I guess the first one was it looked like it had C4 cyclic symmetry. Did you do the 3D variability analysis with that symmetry applied, or did you refine it in sort of asymmetrically and then do the 3D variability analysis?
I believe I'm not 100% sure if the 3D variability was with C4 symmetry, but I know we didn't do C4 symmetry for when we solved when we did the 3D classification, and the final structures are not with any symmetry because we actually see the difference, we actually see the different states in different monomers within the tetramer.
Yeah, right. I guess my follow-up question is, and I guess you hinted at it in your talk, but I guess I'm trying to understand how it was time resolved when you just did a single time step for the UV photocatalysis step. Was the aim to now, I guess, collect more data sets with different photocatalysis time points to actually make it fully time resolved?
Yeah, that's a good point. I mean, I guess that's just a matter of what we call it. I think
Marketing.
we I mean we did capture different states. I think if yeah, we could have used different caging systems, but I think we could have also would have been more clever, would have just been waiting a little bit longer, or having it more UV light on, so we could catch at different steps. But I feel like we already, because of the variability of the sample, we did catch it at different stages. So I think looking back, we should have waited a little bit longer, perhaps, and have captured even a later time step.
Cool, thank you.
That makes
Oh,
sense.
thanks. Thank you. Do we have any more questions from the audience?
Must have been so easy to understand, hopefully.
Yeah. So what's next for this sort of research project? I assume this was in your past lab. So do you see this project heading somewhere in the future? What's
I
the
mean,
next phase?
I know that for the elongasome project, we want to try to isolate it by tagging on the in the chromosome, and so not overexpressing and trying to capture some of these complexes where the protein's expressed endogenously at endogenous levels. For the GtrB, I think the plan is to try to use the same system on other enzymes to see if we can repeat it. I will say that the advantage of GtrB is that it's relatively bigger, or it's a lot bigger than most of the other enzymes I talked about, which it's and more stable. So it's a little bit easier to work with than some of these small enzymes. They're already challenging to solve the structure of
Mm. Yeah.
without trying to capture multiple different conformations. So
Yeah, I can imagine. And also the part of the protein that was more flexible, even though it was in the same sort of transmembrane region, besides the fact that it just kind of moves to capture the binding partner. Do you know why that can be happening? That this bit was more, you captured less of a resolution there?
which one do you mean,
This
the
was,
for the Friday?
yeah, so you had the protein in a transmembrane region and then left
Well,
bit.
I'm thinking it's because they're not we're seeing some movement between the two, and the larger
Mhm.
of the two, we're able to align better. So then we see the flexibility of the other
Of the
not
other
it's
part.
not a subunit, but it's part of the protein, yeah.
Cool. Well, thank you. So do we have any questions before we finish? Just gonna wait if anyone wants to type them in the chat box. But if we don't, thank you so much, again, for giving us the talk. So just on behalf of everyone today, the organizers, audience, and myself, thank you again for joining us here today and sharing your amazing research. And thank you also everyone for joining us here today, and see you at the next seminar. So yeah, thank you and have a lovely day ahead.
Thank you so much. Thanks for
Thank
attending.
you.